Hey, my one students, Mr. Yance again, starting a new chapter. We are headed into genetics, which is one of my favorite chapters I teach. Probably no more important of a chapter to know in biological living systems and how we pass traits on from cell to cell and from one generation to the next. So we're going to look right at the beginning here. We're going to talk about what is the definition of genetics. And I think a lot of people think genetics and heredity are interchangeable words. Technically, genetics is the science of heredity, which means it is the science of potentials, passing on information from cell to cell. As we know from a previous chapter, that could be mitosis if we're making blood, skin, muscle, bone, or it might be meiosis where we're passing that information from egg to sperm cell. Now we're going from one generation to the next. Okay, and what I have on the screen is really important. There's two big questions in biology. What are the two great influences that make you who you are at this moment in time? And that is your heredity, your genetics, and your environment. Now, the question I always ask my students is, which one of those two is more important? Heredity, where you don't have any say in it, but that's a trait you get from your parents, or environment, which is all the outside forces that act on you, the people you hang out with, the places you go, your home life, your school life, your diet, all that good stuff, right? So what it comes down to is, let's just for argument's sake, somebody might say, well, heredity is more important. If you don't have the right chromosomes, you don't have the right genes, then you're not going to become a living thing, which is true, right? Somebody else might say, well, wait a minute, you can have all the best genetics in the world, but if you don't have the right uterus and the right umbilical cord and all this stuff, then you're not going to exist as well. So you can see where this is going. You can't say one's more important than the other. I will say this, and I think all of you will agree with me on this one. I can't do anything about what genes my mom and dad gave me. I was stuck with their hair, eyes, whatever I get. However, I can change my environment. So if I don't like who I am and I don't like the person I become, I can change my friends. I can move away. I can go to different places. I can meet new people. So we do have a little more control over number two than number one. But both of them in the realm of biology are equally important. Okay. So I want to kind of look at the history of genetics. And let me start with showing you a real quick video on a guy named Gregor Mendel. All right.
Monk, teacher, and biologist, Gregor Mendel is known today as the father of modern genetics.
Gregor Mendel's experiments on pea plants were fundamental in establishing the field of modern genetics.
Born Johann Mendel on July 22, 1822, in what was Austria and is now the Czech Republic. Mendel excelled at school and became an Augustinian monk to further his education. He was given the name Gregor when he entered the monastery.
The monastery sent him to the University of Vienna, and that gave him the education in science, math, chemistry, and botany that he then brought back to use in his teaching.
Mendel accepted a teaching position at the monastery in Brno after completing his university studies. It was during this period he began his famous pea plant experiments.
Before Mendel, people thought you would equally inherit the traits of parent A and parent B in equal proportions.
Mendel experimented with pea plants and found that instead there were dominant and recessive traits in all organisms that were passed on statistically based upon the mixing of genes between the two parents.
This theory is now known as the law of segregation. Mendel also formulated the law of independent assortment.
The law of independent assortment addressed the fact that there are a large number of traits. These traits are passed on randomly and are independent of the other traits and of the traits of the two different parents.
The ability to explain hereditary diseases and to engineer plants with desired properties are all consequences of the fundamental theory founded by Mendel.
During his life, Gregor Mendel's research was disregarded by almost everyone. It wasn't until the early 1900s, after his death, that Mendel's work was appreciated and now serves as the basis for genetics and heredity.
In his late
Okay, so that kind of shows you a little bit about who Mendel was. If you look on my screen here, you'll notice that he is known as the father or pioneer of genetics. He kind of started all of it, which we're going to talk about him for just a little bit here. Occurred back in 1856 to 1866. Now, understand he was, a lot of people say, well, he was a monk. What would a monk know about genetics? But he was educated, as you could tell from the video. He went to college. He studied botany and took science classes, and then he went into the monastery. So he was well educated, and he did these experiments. And what's amazing is he did these experiments over a 10-year span, which if you know how impressive that is when I say that, because I have a hard time getting my students to concentrate on 2 days of labs, let alone 10 years, right? So he had a huge volume and information and data he gathered in a 10-year span. So this was not just some haphazard experiment on pea plants. He was well thought out, it was well done, and he was way ahead of his time as far as science goes. So he published his results in 1866. It didn't become really famous until 34 years after that and 16 years after he died, which is kind of ironic. But I don't think fame and fortune, he was a monk. They live a pretty humble existence. But, pretty amazing that he came up with these theories clear back in the 1800s, and we're still talking about him today. A lot of textbooks are named after Gregor Mendel. He is well known in the realm of genetics. And what's amazing is when you're talking about 120 some year span, genetics has exploded in that time span. Okay. And remember that we didn't even know DNA existed until the late 50s, early 60s, 1960s. So, I think I may have said this in class or on a video that my mom, who graduated from high school in 1950, we didn't even know what DNA was yet. And she's still alive today. So it's a pretty young science, but it's a very fast-growing science. And it all started with Gregor Mendel. And it wasn't an accident that he chose a pea plant. Perfect plant to choose. So let's kind of look at why was Mendel successful? Why are we still talking about him hundreds of years later? Why is he known as a pioneer of genetics? There's 3 reasons I want you to know. Good short answer question on the test. One reason was the variety of pea plants exhibit. Not all plants would exhibit the variety that peas do. I just listed a couple of them there, 3 of them, like height, seed color, seed texture. What's impressive is they're very, very extreme. So he could walk out into his garden, he could tell a tall plant and a dwarf plant. It's not like a tall plant was 3 foot and the short plant was 2'11", right? They're extremes: yellow seeds, green seeds, purple flowers, white flowers. You can easily go out and make observations. And remember, Mendel's making these observations and writing them out by hand. No computer, no spreadsheets, no fancy ways of collecting this data. It was the old-fashioned go out and make your observations and write them down, right? So he picked a perfect plant. The other reason pea plants were a great choice by Mendel, and we think this was all by design, because he was known for his botany. He was known for studying plants, right? Was how they reproduce. So a little botany lesson, a little reproduction of a plant. The reproductive part of a plant is a flower. A lot of people don't realize that flowers are pretty and look nice, not so we give them away on Valentine's Day, but because they want to attract insects so they can pollinate, reproduce. So inside of a flower, there's a thing called a stamen. That's these things sticking up on these stalks. They'll produce the sperm, which is in the form of pollen. And the pistil, P-I-S-T-I-L, is this little structure in the middle. It's a tube with a little fuzzy, sticky top to it. And what happens is when plants reproduce, they're going to basically fertilize the egg with the sperm, the pollen. Okay, now there's 2 ways we can do that, right? So basically there's 2 methods. We can just have it come over here on the same plant, and the pollen gets released, it gets stuck, drops in this tube, fertilizes the eggs at the bottom. Or I can have a bee come and get the pollen and go over to another plant and pollinate it that way. So we have some terms for that. Okay, so let me kind of show you that real quick. So we have self-pollination and cross-pollination, okay? And then that sperm will then basically develop into a seed, and then that seed hopefully gets deposited somewhere in the soil and grows into another pea plant, right? Now, he's got two things going on here. He's got a built-in control group, okay? We talked about this in the first chapter. So he's got this self-pollination where pollen is transferred from stamen to pistil in the same plant. So Mendel went out in his garden in a little corner and let his pea plants reproduce like they naturally would. The benefit of that is tall plants are going to produce tall. Dwarfs are going to produce dwarf. Green seeds, yellow seeds. And he had a group that he knew what was going to happen, and it gave him something to compare his other group to, and it also gave him pure breeding stock. So he knew that if I crossed a tall one, I had tall genes. If I crossed a dwarf one, I got dwarf ones, right? So that was his control group. The one he came up with all of his findings with is the cross-pollination group, where you're transferring pollen from one plant to a different plant. And he did that a couple ways. He took a little camel hair brush and got the pollen off of one plant and went over and sprinkled it on another. So if you wanted to cross a tall with a dwarf, easy to do. Yellow seed with a green seed, you could do that. He even took, and this is crazy, he even took out the pistil and grafted it into a different plant and then let it pollinate. So pretty crazy. And this was his experimental group that he could look, say, hey, here's what I'm crossing. Let's see what happens. We'll talk about those experiments in the next video. And then he had his control group that he could actually look off and see doing his thing. Okay? So we'll come back to that. So kind of put that in your brain. Okay? Third reason was just Mendel. He was successful because he was well organized. He was very good at math. He did everything statistically. He kept very accurate data, very a good observer, and is talked about still today because he was very detailed oriented. All right. So that's just kind of an introduction. Hopefully that makes sense to you. If not, shoot me an email, ask me questions. That's perfectly fine. I'm going to go through and show you next video on some of the experiments that he did and how he came up with his conclusions from them. All right. Hey, have a good day, and we'll talk to you later. See you. Bye.